I'm truly honored to get to talk to you about a subject that I have been passionate about for quite a while. I have been fortunate to be a researcher of FASD for over 10 years now. And in the third year of my PhD training, I recognized that I wasn't hearing a lot about paternal contributions to FASD symptoms. And in the course of a couple of weeks, I was able to review the entirety of available literature on it because it was just so scarce. And I'm going to provide a very brief overview of this history. The first study that I was able to find looking at the effects of a father's drinking on offspring was over 100 years ago in 1918, and this was a study in guinea pigs. Almost immediately, research within the United States studying alcohol ended at this time because prohibition came about, and there was no need to study alcohol anymore because nobody is drinking. Even though prohibition was repealed in 1933, we did not see investigations of alcohol use, either maternal or paternal, for still a few more decades, until the 60s, which is what then led to the first clinical description of fetal alcohol syndrome in 1973. But we were still seeing the predominance of work was investigating maternal exposures. It was really only in the 90s and early 2000s that we started to see a small rise in paternal alcohol contributions to offspring health, and this was led by researchers like Gregory Homanix and Ernest Abel. And what I found from reviewing this literature was that we were seeing a lot of the symptoms that we usually ascribe to maternal alcohol exposure. And this included lower fetal birth weights, changes in organ development. In preclinical models, we saw more runts were born to these litters. We also saw changes in physiology, like increased cortical thickness, reduced testosterone levels, as well as neurotransmitter changes. Behaviorally, we were seeing changes in spatial learning. We saw more anxiety and impulsivity, as well as heightened stimulant sensitivity, which was one of my earlier indications that maybe we would also be seeing changes in how individuals are seeking substances themselves. And together, this all really feels like an FASD-like phenotype, but it has been seen in cases where maternal alcohol exposure was not present. The alcohol field, I think, in some ways is really behind with respect to looking at this. There have been other fields that have shown that paternal experiences can influence offspring. So we've seen that chronic stress before conception can make offspring more stress sensitive and anxious, even when their fathers were not there to raise them. We've also seen that a high-fat diet or a nutrient-poor diet can lead to changes in metabolism and weight difficulties in children as well as grandchildren. We've also seen that exposure to toxins like pesticides and air pollution can be linked to altered brain development, and even other recreationally used substances like nicotine and cocaine have been shown to change reward behaviors in their young. And so this all does lead us to suggest that there are some molecular changes that are being inherited and that are impacting offspring health. And it's very tempting, and as my mentor Rajesh Miranda would say, it's rather low-hanging fruit to assume that these changes are due to alterations in our genetic code, in our DNA, since fathers don't gestate or nurse. This has been investigated specifically with respect to the heritability of alcohol use disorder, or AUD. However, these studies have produced really surprising findings. Through twin and adoption studies, it has been estimated that the heritability, the genetic contribution to alcohol use disorder, is about 50%. So this is completely eliminating environmental influences. However, when you look at changes in DNA sequences, for instance, you expect to see a T, a thiamine, but instead you see a C, a cytosine. Those kinds of changes make up less than 1% of the contributions to AUD heritability. It really seems like there are a lot of factors that are being inherited that have nothing to do with an exact change in DNA. And this is what we call missing heritability. What else is being inherited and affected by paternal exposures? So for a long time, people assumed that sperm only carried DNA, like an instruction book, and that it did not contain anything else. But we know that sperm also carries chemical information that guides very early periods of development. And one of the best studied factors is referred to as DNA methylation. And these are simply tiny chemical tags that act like on and off switch for whether genes are expressed. And notably, after fertilization, an embryo performs a massive reset of these on-off switches, but some of them are maintained between father and child, and then they're copied into the child's cells as they divide, and so those can be maintained. There are also these proteins that are called histones that wrap around DNA, and in humans, up to 10% of the histones from a father are transmitted to their child. So maybe it's not the DNA itself, but rather these proteins that are hanging out with the DNA that are leading to changes in outcomes. And finally, there are these tiny RNA that can, during the first few days after conception, change whether genes are turned on or off. These are also inherited. And during these really early periods where so much is developing, this could absolutely be sufficient to explain heritability. But right now, we just don't have enough research to say for sure if alcohol is affecting these mechanisms. I want to switch gears here to talk about some studies, and I'm going to skip this one for right now for time's sake, to talk about studies that have looked at paternal alcohol exposure, and particularly its contributions to FASD. So this study was published a little less than a year ago. Many of you may recognize the researchers who were involved in this project. This was a secondary analysis, meaning that initially this study was looking at FASD among 5th grade children in the Western Cape province of South Africa. And in the initial studies, they were just looking at maternal exposure impacts. And then they went back and they found reports of paternal alcohol exposure from these cohorts. And so they went on to publish this analysis to say, we know individuals within these cohorts expressed FASD. If there were reports of paternal alcohol exposure, did that increase the likelihood of FASD symptoms? And if so, which symptoms? And so I highly encourage you to read through the paper. I want to highlight the biggest takeaways from this for the sake of this presentation, which is that in this analysis, the researchers did something we haven't seen in human studies before. They grouped families by whether a mother alone, a father alone, both parents, or neither parent used alcohol before or during pregnancy. And what they found was that children of fathers who engaged in binge drinking, this is 5 or more drinks within a session, tended to be shorter in stature and have smaller head circumferences. They also found that there were cognitive impacts. These children tended to demonstrate lower verbal intelligence scores. And notably, even when they accounted for whether a mother had used alcohol or had tobacco exposure, these effects still persisted. This effect is driven by paternal exposure. I do want to note for transparency that when they compared the degree of effect of the paternal exposure to the maternal exposure did have a larger impact. However, in the instances in which both parents had a history of alcohol exposure, these effects were the most severe. And so this study is really impactful for being one of the first to demonstrate that 2 parents together can produce distinct outcomes when they have a history of preconception exposure. And this was really meaningful to me because of what we know of drinking patterns in human populations. And so here I want to introduce a phenomenon that's called the partner influence hypothesis. It's a very simple premise. It's the idea that the people closest to us shape how we behave. And this includes how we drink. In romantic situations, individuals may start out with different preferences for how much they drink. But over time, cohabitating together as well as social expectations, they will eventually develop more synchronous drinking habits. And I want to mention several studies have demonstrated that this is not gendered in heterosexual couples. Women can influence men's drinking and men can influence women's drinking as well. It's more of the idea that they will synchronize. And what that means is that in our understanding of FASD research, where we have demonstrated instances of maternal alcohol exposure, it's very likely paternal exposure is also happening. And unfortunately, right now, asking about paternal alcohol exposure is not a standard and consistent factor in our analyses. And as a preclinical researcher, it is certainly not being represented in FASD models to the degree it is likely happening in real-world cases. And so this led to one of my first questions, where I wanted to determine whether or not preconception alcohol history in a dual parent model could be associated with distinct outcomes from single parents, as the Cape Town, South Africa cohort had demonstrated. And for this, I wanted to use a fantastic, publicly available database called the All of Us Research Program, which is led by the NIH, and it is a research database designed to understand how biology, lifestyle, and environment shape health. It includes data contributed by hundreds of thousands of participants across the United States. And importantly, it includes a range of data, including health surveys, electronic health records, physical measurements, Fitbit data, as well as genetic data. And together, I can look at these factors as a variable of interest, family history of alcohol use disorder, which was one of the survey questions provided to participants. And as this question was posed, including yourself, who in your family has had an alcohol use disorder, these were the options available to participants. They were able to separate whether their mother or their father had an alcohol use disorder, as well as a sibling, a child, or a grandparent, and they could make multiple selections. And this was really exciting for me to see this variability because it meant that I could simulate that same dual parent AUD exposure. For the purposes of what I'm sharing today, I was really interested in looking at whether or not dual parent alcohol use disorder diagnoses would increase their child's likelihood of either receiving their own AUD diagnosis or engaging in more drinking. And so for this cohort, I selected adults who completed alcohol use survey answers and provided their family history of alcohol use disorder diagnosis data. These were derived from electronic health record, diagnostic codes, ICD classifications for alcohol dependence, misuse, or use disorder. And then separately of the electronic health records, we had the individual survey data, where individuals reported how much or how often they drank, and these were the options available to them, as well as how many drinks they would have on average within a session. And I want to make sure to caveat here that while it's easy to assume that individuals who drink more often also drink more within a session, we know that people have a variety of drinking patterns that they engage in. Alcohol researchers who specialize in study adolescents will often see that they don't drink often because they don't necessarily have the availability, but when they drink, they will drink considerable amounts. And so we don't want to conflate these. These are two different measures. And importantly, having survey data separate of a diagnosis is really valuable. I don't need to tell the people in this room that gaining a diagnosis is not always easy or desirable for individuals. And so having that as a separate element, I think, is also powerful for this particular cohort. My analyses were respectively regressions. I want to point out a very important caveat with this cohort. As I'd shown you earlier, these were the options available to participants on their survey. They did not have the option to elect that nobody in their family had an alcohol use disorder. I'm hoping that those who have worked with survey data will empathize with me on this. Unfortunately, this means that I do not know if the people who didn't answer this question truly don't have someone in their family with alcohol use disorder, or they didn't see or understand or want to answer the question. And so instead, my control group consists of people who had a family member with alcohol use disorder that was not their parent. And so this group will already have a family history with AUD. I have two models that I'll be sharing with you today with respect to looking at parental effects. The first one I call my strict model, Model S, and this means that for our parental groups where mom, dad, or both parents had an AUD, no one else in the family could have an AUD. I excluded that. The only person who could have it was a parent. This hopefully allows us to minimize any contributions of other family members' history of AUD, but we have to recognize that because AUD is heritable, you're also probably eliminating a lot of the population when you do that. And so I also have my Model I, which is my inclusive model, which does include those individuals. And so I'm going to be sharing both of those with you today. And so here are my results. This is our first study looking at how parental AUD is associated with the risk of offspring having alcohol use disorder. And what I'm showing you here is our groups divided out. You will see the sample sizes listed here, and as I said, this is a very large data set. So I was also able to separate our groups by females, which are the numbers on the left, and males, which are on the right. And for those of you who are not familiar with reading odds ratios tell you how likely an event is to occur. The number 1 indicates that that event is not more or less likely to happen. It is the same likelihood. The lower from 1 the number is, the less likely it is to happen. And the higher from 1, the more likely it is to happen. And what you will see within our model, where the only people who could have AUD was a parent, there was no effective paternal exposure on the likelihood of a child's AUD diagnosis. However, with maternal exposure, there was a significant but small increase. And notably, in dual AUD, we see an even larger augmented increase. What is notable here is that when we incorporate our inclusive model, so this means family members with AUD could also be included in this model. You'll notice our sample sizes in some cases double. These patterns are largely persistent. We still see an increase in AUD diagnosis with maternal AUD. Notably, the paternal pattern appears, and I think that's really meaningful here, where paternal AUD does increase risk of AUD diagnosis, but you need to ensure that other family members with AUD are also incorporated in this model. And notably, we see an odds ratio of 2.2, meaning that if you have 2 parents with an AUD diagnosis, you're more than 2 times more likely to have that diagnosis yourself. And in our final column, we have our sex interaction. This allows us to see, are there any differences in whether this impacts men or women, sons or daughters? And we don't have a significant difference here. And you can see that in this chart here, where men overall are more likely to receive an AUD diagnosis than women are, but you'll see that the pattern of exposure effects is similar between them, where our lowest group is our controls. Single parent exposure, blue for fathers, red for mothers, increases it, but the highest likelihood of AUD comes from having 2 parents. And so that was our diagnoses, but again, diagnoses are not a full picture. Let's take a look at our survey data. So these are the same subjects, and once again, I was interested in looking at, does parental AUD increase how much you drink, how often you drink, I should clarify. And the results were surprising. It doesn't really affect how often you drink. If anything, there is a small reduction in the frequency of drinking among this same cohort. And that was quite surprising to see. It does seem that there is a slightly greater effect on men. And when you look at our control groups, that might be because within our controls, they are more likely to drink, in this case, 2 times a week. And yeah, it seems that exposure's reducing their frequency. So I went on to the final survey question, which is, how much are they drinking within a session? And then the numbers returned to look like what we saw with our AUD diagnoses. What we're seeing here is that parental exposure is increasing how many drinks they're having at a given time. time. And if you look at our dual exposed model, you will see in men and women that they are engaging in having more drinks at a time. This is 3 drinks per occasion and 5 drinks per occasion, and this effect persists. So what this suggests to us is that dual parent alcohol use disorder is increasing episodic drinking, which isn't necessarily frequent, but when they drink, they're having greater volumes. And this is associated with them being more likely to have an alcohol use disorder diagnosis themselves. I want to be very clear here about how this translates. And as I mentioned, these controls do have a family history of alcohol use disorder. They may not be representative of individuals who do not have AUD within their family. I also want to point out that for the sake of this conference, this is more of a clarification than a limitation. Parental AUD does not guarantee that preconception or prenatal alcohol exposure occurred. That said, it is likely because we know that the majority of individuals who are diagnosed with alcohol use disorder have their first full drink before the age of 17. 2/3 of individuals with AUD are diagnosed by the age of 25, and the current rates of mean age for having your first child is also in your mid-20s. So it is very likely not only that these diagnosed parents engaged in preconception alcohol, they were likely doing so for several years before these data were collected. Finally, I have not investigated contributing factors yet related to socioeconomic status, polysubstance use. Age was consistent across our groups, but we have the capacity to break that down. And I hope that I will be able to share that with you very soon. For future directions, I'm really interested in looking at factors beyond childhood alcohol seeking. I want to look at co-occurring diagnoses. Does paternal exposure lead to increased risk for other symptoms we know are associated with FASD, or co-diagnoses such as ADHD? And I really want to incorporate genetic data. I'm so excited to be able to look at that because I'm going to be able to pull from that minimal literature of paternal exposure and ask questions such as, does dual parent alcohol use disorder history interact with variations in genes that have been associated with paternal inheritance, like brain-derived neurotrophic factor? And does this predict episodic drinking? These are the kind of questions that allow me to go back into preclinical models and see if I can replicate these effects for the purposes of targeting mechanisms. And with my last little bit of time, I am going to just briefly touch on what I'm doing in the laboratory right now. So I am a preclinical researcher predominantly, and I feel it is my responsibility as an FASD researcher to take all the wonderful complexity that makes us human and very consciously eliminate it within the laboratory. Control it, I should say. So we know that in humans, FASD symptoms can be dependent on a variety of factors. This is a non-exhaustive list. And so within preclinical models, we can keep all of these things constant, manipulate 1 or 2 variables to try to establish more of a causal relationship. Am I capable of replicating the effects that we're seeing in humans when I have accounted for maternal age and stress and housing conditions simply by manipulating whether or not one or both parents were exposed to alcohol before conception? And that is what I'm looking at right now in collaboration with Dr. Michael Golding, who we'll be bringing up momentarily. He's really been the pioneer for this within the preclinical field. And I was just fortunate to be able to work with him and his recently graduated PhD student, Destiny Derrico. And within this model of exposure, rodents receive prenatal alcohol exposure, preconception alcohol exposure. They are randomly assigned to receive either water or ethanol. Within our males, they are exposed to alcohol for at least 2 spermatogenic cycles. These are the formations of sperm. And this translates to a man who is drinking casually and then eventually has a child. And for our pregnant females, they receive alcohol for 10 days before before conception, as well as 10 days after. And so in humans, this would translate to a woman who is drinking, discovers she is pregnant toward the end of the first trimester, and then stops drinking. No exposure happens after this point. And this allows us to have the same 4 groups that we had for our prior studies, those who have no parental alcohol exposure, those where a single parent was alcohol exposed, and then finally those where both parents were. And so within my particular studies, trying to compare this to the All of Us data I just showed you, I wanted to see, do these offspring show changes in how much they want to drink alcohol? And so I use operant chambers for this, in which mice have to press a lever in order to receive a drop of alcohol. And notably, across these studies, I can change the demands of how hard they work. So it's not enough to simply consume a lot of alcohol. You, in order to receive an AUD diagnosis, usually need to demonstrate that it is an imposition on your life, that you are craving it, that you are thinking about it. And so we can change how hard we make them work for this. We can measure craving by not giving them any and seeing how hard they try to gain it. And what I'm going to show you today, since these data are only about 80% of the way done so far, is which groups, as of right now, are drinking the most across these paradigms. So I've separated males and females out for you here again. And as of right now, across different conditions, we are seeing that those who were born with both parents having preconception alcohol are drinking the most. And we're seeing this in males and females. So we are replicating what appears to be the pattern within human data as well, which is of course exciting because again, this means that we can begin investigating mechanisms and hopefully translating it back to humans. From here, and where I would like to see research go, and really kind of the takeaway of what I'm trying to give for this particular presentation, is that we need a lot more research looking into fathers and fathers' history of alcohol seeking. We need to study how alcohol alters sperm epigenetics and RNA content so we can understand how preconception alcohol is inherited. We need longitudinal studies that follow children who are born to fathers who drank alcohol before conception. And finally, we need to integrate paternal alcohol screening as a part of FASD standardized assessments. And for preclinical researchers, we need a lot more studies that incorporate both parents. Our ability to be able to compare dual parent models to single is a power and controlled environment that we can facilitate and is going to translate to real world cases of exposure. And finally, I just want to say the big picture of this is that fathers matter. Including fathers in FASD science will allow us to improve how we can predict risk, provide targeted interventions, and with a holistic family approach to caring about FASD, investing in families starts with investing in parents, and that includes mothers and fathers. I want to conclude my talk at this time and just thank you all so much for being here today. I have a fantastic mentor without which this work would not be possible, Rajesh Miranda. I've also been fortunate to have funding from NIAAA, without which this work would not be possible. I'm a member of the Texas FASD Network, which is an incredible group of advocates for FASD, and I'm so honored to have the opportunity to work with them. Finally, I want to shout out to an exceptional group of undergraduates that I've had an opportunity to work with over the last 5 years. These individuals are hardworking future medical providers and researchers, and importantly, they are now FASD-informed citizens who get to move forward and advocate for FASD. In the top here, you will see 3 of my assistants. This was a year ago in March, advocating for the FASD Respect Act with our Texas congressional members. And one of them, Tia Pandey, is here today. She will be presenting our work on prenatal alcohol and cannabinoid co-exposures tomorrow. And if that interests you, please do feel free to attend her talk. Thank you all so much for listening.